Okay, I started recording. Go ahead and go whenever you're ready.
Okay. Moving on to my second article, it is about the ACTG1 and its regulation in intervertebral disc degeneration via the NF-kB, P65, and AKT pathways. As we know so far, the ACTG1 mutation leads to hearing loss in humans. The knockdown of ACTG1 suppresses the proliferation and migration of tumor cells. However, its role in intervertebral disc degeneration, or IDD, is yet unclear. The bioinformatics methods have revealed that the ACTG1 might be a hub gene in IDD. It is, if you're wondering what IDD is, it is the primary reason for lower back pain, which leads to a tremendous economic burden worldwide. The intervertebral disc is composed of gelatinous nucleus pulpus, or NP tissues, and surrounded by annulus fibrosus. The NP cells produce an extracellular matrix, which mainly consists of collagen 2 and aggregatin. In the process of IDD, the production of water content and protoglycan are reduced, and matrix catabolic enzymes degrade ECM progressively, which leads to the destruction of the disc structure. Furthermore, the expression of ACTG1 is severely generated. The nucleus pulpus tissues was low compared to that in mildly degenerated samples. Moreover, the ACTG1 level was negatively correlated with human disc degeneration grades. The low expression of ACTG1 also found in degenerated NP tissues in the rat. So in this experiment, it will be talking about both human and rat as test subjects in various tests and experiments. To further explore the function of the ACTG1 in IDD, the gene expression was depleted in human NP cells via the sRNA transfection. The abylation of ACTG1 increased MMP3 expression, but decreased the level of collagen 2. Excessive adipopsis was observed in ACTG1 knockdown groups, indicating that the absence of ACTG1 exacerbated IDD. The GO function and pathway enrichment analysis for differentially expressed genes, or the DEGs, of two microarray datasets, the GSE 56081 and the GSE 42611, indicated that inflammatory response plays a crucial role in IDD. Interestingly, in the protein-protein interaction network, ACTG1 is connected to the proteins of inflammation-related pathways. Furthermore, ACTG1 knocked down unregulated P65 level, but suppressed PAKT expression. These data collectively demonstrated that ACTG1 regulated the development of IDD through the NF-kB-P6F and AKT pathways, and ACTG1 may be a novel marker and therapeutic target of IDD in the future. In the so sorry, I just came up being sick, or I'm still sick, I guess. So in the results, identification of differentially expressed genes and functional enrichment analysis was discovered. Under the criteria of log2FC over 1 and P, and the P test as in the 0.05, a total of 2,951 DEGs were identified between human degenerative NP tissues and non-degenerative NP tissues in GSE 56081, whereas a total of 212 DEGs were found in the degenerative rat disc as compared to the normal rat disc in the GSE 42611. Sorry. Results have demonstrated that the extracellular matrix organization and inflammatory response played an essential role in IDD, which was consistent with the previous studies in order to identify additional candidate genes involved in the IED. PPI network and ENCODE modules were constructed by Metascape and visualized by Cytoscape software. Complete PPI network of common DEGs was provided as a supplementary file. It was found that the enriched Go terms in Module 1 and Module 2 were also related to inflammatory response. Interestingly, the ACTG1 not only has the highest degree score of 5 points, but also was connected to inflammatory factor of the NFKBIA in Module 1. Thus, the ACTG1 may be a new biomarker of IDD and hence was selected for subsequent study. Next slide, please. So the figure on the left was from my first article, and it shows the stats of the things such as the growth analysis of the MFEs in figure A, or the cell size or viability in both B and C. And it is the comparisons of the null versus the active ACTG1 in rats and humans. And then my figure on the right is from my second article, and it's showing the difference between the control group and the IDD group of rats and the amount of ACTG1 positive tissue cells in either the control or the IDD group, as well as the histological score. As you can see, in the ACTG1 positive, the ACTG1 positive cells were higher in percentages in the control group than the IDD group, but the opposite was said for the histological score. Next slide, please. And these would be my citations. The bottom one is for the so sorry, is for the the first one, and the top one is for the second one. Thank you.